So let's go ahead and continue our discussion about the cell cycle and how cells divide. Keep in mind the whole cycle, if we're talking about skin or hair, various body cells, the whole cycle generally takes 24 hours. The bulk of that time is spent in interphase. And then when the cell gets to mitosis, that's when all the activity happens, when we can actually physically see the cell going through these different phases of the cycle. We talked about it in the last lecture. Mitosis starts out with prophase. This is when the cell takes the DNA, assembles it into chromosomes, nuclear envelope dissolves and breaks down, and it says, all right, it's time for us to start moving towards the rest of this process of mitosis. So after prophase, the cell then kind of progresses into prometaphase. This is kind of a weird part of the cell's cycle. It sits in between prophase and metaphase. The main things we're going to see here are chromosomes start attaching to these little strings, what we call microtubules, and those strings are going to be anchored to the little green, will look like little green star-like structures in this diagram. And those are known as the centrioles. So the centrioles are the anchors of the animal cell that holds a string that stretches out to the chromosome, and that helps the chromosome line itself up along the center of the cell, or you'll see it called the equator, the center line of the cell. Now, please remember or keep in mind when we talk about the equator or the line of chromosomes, it doesn't have to be left to right. It could be top to bottom. It could be on a 45-degree angle, 30-degree angle, or whatever. It's not a flat cell left to right line of chromosomes when we're looking at this. It could be in any angle, but the idea is the chromosomes line up across the entire cell. They form a line during the beginning part of prometaphase. As they progress into metaphase, the line becomes much clearer. Now you look at this line, and it is not left to right. It's not even top to bottom. It's on a slight angle, and that's completely fine. But notice the orientation of the chromosomes. They're all lined up across the center, but they're stacked end to end. That way, the center of the chromosome, the center of the X, it's always going to be facing those centrioles. And if you look at the little green lines, what we're calling the kinetochore microtubules, those lines attach to that center of the X, the center of the chromosome. So metaphase means middle. Chromosomes are lined up in the middle of the cell, and they're attached on opposite poles with these little microtubule fibers. They're also referred to as spindle fibers. So prophase is first, then prometaphase, then we get into metaphase, and after we've lined everything up, the cell progresses into anaphase. Ana means apart. The chromosomes are being pulled apart. Now you could call each half an individual chromosome. You could refer to them as chromatids. Terminology gets a little fuzzy here, so I would really highly encourage you guys, make sure you look over the terminology and understand how much genetic material is where at any given point in the cell. I like to keep it separated with terminology. When the DNA is all mixed up and looks like a big pile of string, chromatin. When they form the X, I call them chromosomes. And then what I will refer to these halves as, I call those chromatids. You could call them sister chromatids if that's more appropriate for you. But I generally avoid calling these little halves chromosomes, like what they've done here in the picture, just because it gets confusing. So again, my terminology, what I think keeps it simpler, chromatin is mixed up noodles of DNA. Chromosomes are X-like structures, and chromatids are half an X, or what looks like a little V. Whatever you want to call it, during anaphase, the genetic material is ripped in half. Ideally, half goes to the right side of the cell, half goes to the left side. The halves move to opposite sides of the cell. Doesn't matter if they're going right and left, top to bottom, 45-degree angles each way. They're moving apart from each other. The other main thing I want to mention that happens during anaphase is, and look for the arrow here, is this happening here. Notice the indentation, this kind of divot or depression happening in the cell membrane. Same thing on the top here. So a process known as cytokinesis is actually starting during anaphase, and that is when the cell starts getting squeezed or pinched in half, if we're talking about an animal cell. Because animal cells are soft, they have membranes, and they're getting squeezed in half by a ring of protein fibers. These things we'll call the contractile ring. Plant cells, they work a little differently, and I'll show you the difference in a few minutes here. So, prophase, prometaphase, metaphase, anaphase, and then the cell progresses into telophase. Now during telophase, the cell continues squeezing in half. So these things are all happening all at the same time. It's not a start this, stop, then go to this, stop. It's kind of this gradual, continual process. So during telophase, half the genetic material has gone to one side of the cell, half to the other side of the cell. That genetic material starts to unravel or decondense itself, as the term describes it here. So instead of having these little V-like structures, again, I call the V's chromatids. Those chromatids start to just unravel and turn back into that pile of what looks like spaghetti noodles or strings we call chromatin. So as those unravel, new nuclear envelopes will form around each pile of chromatin. Those little centrioles, little green anchors, those are going to dissolve and break down, and new things like Golgi apparatuses and endoplasmic reticulum, they're going to start to build and form. All this is going on during telophase. It's a progressive process. It may take an hour and a half, maybe even up to 2 hours. So it's going to happen over the course of that entire 2 hours of the cell being in telophase. Telophase is officially over when the cell finishes the process of cytokinesis. Now, cytokinesis is the physical division of the cell. So pinching the cell in half, or cutting it or cracking it in half in the case of plant cells. That is what cytokinesis is. It's breaking or cutting that cell in half, not the DNA, not the genetic material. So the flow chart of the process here of mitosis makes it look like telophase ends and then cytokinesis begins. That's not accurate. Cytokinesis actually starts during anaphase and is squeezing or pinching the cell while it's going through anaphase, as it continues through telophase, and then again, kind of that signal to say, telophase is over, boom, we're done. Cytokinesis finishes, and you have two cells side by side that are exactly the same, except they're half the size of the original. Because you took one cell and pinched it in half, now you have two smaller cells. In animals, the structure that pinches it in half is called the cell or the contractile ring. As the ring squeezes, it creates this depression we're looking at here that is labeled the cleavage furrow. So the contractile ring is a structure, the cleavage furrow is the depression or that indentation that forms. Okay, so again, cytokinesis, make sure you write it down, fill out the lines here. Cytokinesis is the physical division of the cell body, the cell structure. So here's our contractile ring. That is the structure that will squeeze that cell in half. So imagine putting a big rubber band around a water balloon, and that rubber band getting tighter and tighter and tighter and squeezing it in half until eventually it pinches the cell in half, and you have two cells or two water balloons sitting side by side next to each other. Okay? And that's the contractile ring for animal cells. In the plant world, plants use a structure called the cell plate. Because remember, plant cells have cell walls. They're hard, they're rigid, they're full of cellulose. It's a good polysaccharide to remember for plants. Cellulose, or roughage. That cannot get squeezed in half. You can't pinch a cell wall in half. So instead, tiny little bags, these little vesicles, start lining up and stacking on top of each other, end to end to end to end, across the length of the cell. And it starts during anaphase, continues during anaphase, all the way into telophase, and by the time telophase is over, you have a complete cell plate that has just cracked the cell in half. So everybody listening to this lives in Illinois, most likely. You guys are near a cornfield at some point. There's a saying about hearing the corn grow. If you sit near a cornfield in the middle of summer and you hear those little cracks and those little pops, the little cracking, popping noise, those are the cells that are cracking in half. Plants use a cell plate, and when those little vesicles line up and create one big long line, they eventually crack and pop the cell in half. Now, it doesn't physically separate and shoot off and go across the cornfield, but it cracks and it creates two cells side by side that are identical to each other. The only difference, they're half the size of the original cell. Same scenario as what we saw in the animal cell. Half the size, cut in half or broken in half. So a little bit of a difference between plant and animal cells in regards to how they actually physically divide. Now, once the cell is finished with telophase and cytokinesis, because it kind of ends simultaneously, the cell then goes all the way back to interphase. Starts interphase over, goes into G1, and says, hey, G1 means I'm supposed to grow. Both cells side by side will start to enlarge. They will double their cell organelles. They will increase the cellular components. It goes into the S phase. It says, oh, we only have half our DNA. Let's make a copy of it. So it will double the DNA, make a duplicate copy of the genetic material during the S phase. After it's done that, it goes into G2, and that's where it takes genetic information, uses that to assemble proteins that will be needed during mitosis. And then it's back to mitosis again. So if you started watching one cell in your body, one skin cell, on Monday afternoon at 1 o'clock, by Tuesday, you have 2 cells. By Wednesday, you have 4. Thursday, you're up to 8. And just keep doubling every day of the week. And these cells are going to spin through this cycle roughly 50 times until the cells say, you know what, we're done. We're old, we're worn out, and we're now finished. So let me make a note here. 50 times. And then the cell dies. It goes through this process called apoptosis. That is normal, natural process. We want the cells to die after so many turns through the cycle because it means we're done, but they've left behind lots and lots and lots of copies. We'll talk about cancer, and the problem with cancer is those cells never die. They never go into apoptosis. They never stop dividing. It's never going to go away.